Welcome to solverfy.
How do individual genetic maps relate within individuals of one species, and what can it be used for? So let's take a case, uh, ourselves, humans. We are 99.9 similar in our DNA sequence. 99.99 similarity. So that means that out of the billions of nucleotides that make up the human genome, 99.9% will have an identical sequence. That means that a difference of 0.1% accounts for all the physical differences in humans, be it eye color, height, metabolism, anything that you can measure is accounted, and that is under genetic control, is accounted for 0.1% difference. Um, Now, you want enough difference in the genes of different people to have genetic variety, so you have different alleles in order to adapt to different kinds of environments that the species may be involved to. But you want the genes to also be similar enough so that when two cells from two different individuals come together, they can form a coherent genome that can be replicated and that can be transcribed. So there's sort of like a sweet spot that you want enough differences to have diversity, but enough similarities so that you can form a functional organism. Okay, so let's look at our answer choices. Individual genetic maps in a given species are genetically similar. Yes, they are genetically similar. In fact, in humans, they are 99.9% similar. Genetically identical. No, they're not genetically identical. If all humans were genetically identical, we would all be clones of each other, right? And we're not all clones of each other. Choice C are genetically dissimilar. If they were genetically dissimilar, you couldn't form a functional human when two people make a baby. So choice C is not correct. Choice D, not useful in species analysis. And that is also incorrect because it is very useful in species analysis. For example, suppose that we are looking at a species and we see 99.99% similarity. That means that there is very low diversity in the genetic makeup of these species. That means that they are going to have inbreeding problems. So you can perform this population or species analysis by figuring out how similar the genomes of different individuals are within a species, and basically make a decision that, okay, this species has a problem because they are too genetically similar, and genetic problems are bound to arise. Because if they're all so similar, if a genetic problem arises, it's basically going to be in all of them, right? On the flip side, and these are hypothetical numbers that I'm making up, okay? But on the flip side, let's say you look at a species and it only has 95% similarity. And for a species, this would be on a low end, okay? So if you have a group of individuals and among them there's only 95% similarity, well, you might have a case where this species is evolving into two different species. You have speciation happening. So deciding the similarity or how individual genetic maps are related within a species is actually very helpful in analyzing species populations. So they are useful, not useful. So choice D is incorrect. Choice A is the only